Okay, so hello everybody, welcome to our 5th session and last in the series of this webinar series. Today's speaker is Stacey from our Singapore lab, and she will be talking about data integration, batch effect correction, and multi-omics data. So in this webinar, we will discuss the causes of batch effect occurring in single-cell RNA sequencing data and how to remove them. Also, the different methods for data integration coming from multiple layers of information and their pros and cons. So in summary, we will cover what is batch effect, how it can be removed, and what is the data integration in multi-omics. So the talk should last about 45 minutes, and at the end, we will have a Q&A session for around 15 minutes. So if anybody has any questions, you can either raise your hand. At the end, I will unmute you and you can ask your question, or you can write your question in the chat and I will ask it to Stacey at the end. So over to you, Stacey.
Thank you, Jennifer. Thank you to the audience who has been with us for the 5 webinars. It's nice to see you all here. So today I'm going to cover introduction to the batch effect in the single cell, mainly focus on the RNA-seq analysis. Yeah, so like nowadays, as unavoidable and due to the raising popularity of single cell experiments, we have been seeing more and more single cell data that is available either in public or in databases like SynEcosys from SingleRound. Or the some of the repositories. So a lot of times as us researchers will need to collect multiple sets of data from single cell experiments and then conduct the downstream analysis. So today I'm going to cover the few topics, like, for example, I will start from what happens when the batches of data are collected and analyzed together. Is it okay to just analyze the raw data, or do we have to conduct the batch corrections when integrating multiple data sets? Like how to observe or evaluate batch effects, what methods are currently available now for batch correction analysis. And we did a small benchmark with the 7 tools that is available for the batch correction methods, and then propose some of the metrics that we can look at with regard to the observing the batch correction results. So at the end, we are going to show a case study with batch corrections to find which we use from a map batch that help us to find a prognosis cluster with the disease of multiple myeloma. So without further ado, let us quickly look at the topics, like what happens when the batches of data are collected and analyzed together. So in the single cell sequencing, batch effect sometimes is unavoidable due to like lab processing, different reagents used, technical variations, or even like a different days of experiments, or the multiple lens in the experiments. For different platforms, they may collect different genes, and the dropout rates with regard to different platforms or different sequencing experiments, they can vary. So the batch effects may well happen when multiple single cell data sets are consolidated together. So as I believe that we have all heard from Shijing's 3rd webinar, where she talked about the standard analysis pipelines. So one of the part of the standard analysis comes from the part where the single cell experiments went through the UMAP reduction, where reduced dimension are generated, and the principal components analysis was conducted or calculated from single cell experiments, and the clusters was calculated based on these reduced dimensions. So if the batch effect happens, these differences in the batches from the technologies variations, it may well hide the true biological differences. For example, the top right figure, it comes from one of the posters from our collaborator, Dr. Chen Han. where he did a study with map batch, like on the right side. The UMAP here are colored by the batches or the different samples, and on the left side, the colors are based on the cell types. So as we actually want to observe from the different experiments, we want the biology to stand out. We want to hide the batch differences as much as possible. So we are expecting from the left side, for example, all of the close colors, they should actually be located at approximately similar locations, so that later when the clustering was calculated from PCA, for example, can return meaningful results, that the clusters will be mostly driven by the cell types, instead of here on the right side, we observe a lot of batches here, that they are, for example, the blue color batch obviously is like co-located together, and the difference in the technical differences may stand out instead of the cell type differences. So how to inspect the batch effect and make sure and observe, for example, the batch effects actually happens in my data. So one of the intuitive way is to inspect the UMAP dimension reduction and check the clusters returned from the reduce the dimensions. So like from both examples on the top right and the bottom right, so both these two examples shows obvious batch effects from a look at the UMAP. And then there are some quantitative methods to evaluate the batch effects as well. For example, the systematic batch effects can be observed from the PCA correlations. And but not all the time the batch effects come systematically from all the experiments or from all the genes. Sometimes we, from our own studies or research before, the batch effect, it can also come from a particular gene. When this happens, this normally happens when the different platforms have different capture capabilities for different genes. So when this happens, that the batch effects only exist in the particular gene, by removing those genes from the expression matrix, sometimes it can resolve the problem of batch effects already. And also, yeah, sometimes the batch effect may happen when the selected batches has the effect. So the batch effect is not systematically appear in all of the batches in the experiment. So actually all of these methods can be inspected, and then the batch effect tools may take care of them when conducting the integration of the data. So the batch effect may result in two problems. First, as we mentioned just now, the subgroup of cells, which is represented by the calculated PCA based distance calculated clusters, this may be highly influenced by the batch effects. So when we do any differential expressions between clusters, we find that the differentially expressed genes that is due to technology differences, but we lose the visibility of the differences, for example, between the responding versus non-responding patients in the same cell types. So regardless of this, sometimes the batch effect is not 100% necessary. Sometimes we do skip batch correction if there's no strong batch effects observed, like intuitively, for example, if the UMAP returned from the initial analysis, it shows like all of the green colored cells are already co-located at the same location. Taking like a multiple myeloma, for example, if all of the T cells, they are located at the same location, but we only observed the plasma cells at all the different locations, it may indicate that the batch effects here in the experiments are not so obvious that we may just continue with the raw experiments. Okay, so here we did in order to show the results or examples of batch effects and how to correct them, we did a small benchmark. just to cater for this webinar. So it combined the data sets from the experiment that all of these data sets comes from CIRAD. We combined the about four different sources of data. So it included the bone marrow data from CytSeq, which include the CytSeq information, expression assays from normal bone marrow, and another 3 different data sets using either normal bone marrow or normal PBMCs. And then I think the fourth one is one of the popular data set from CIRAD that are the normal PBMC data comes from multiple platforms. And the last one is the stimulated and control PBMCs from a patient. So all of these example data sets, we did initial quality control to remove the potential doublets with a high number of feature RNA, and removed the potential debris with a lower feature RNA, and removed the potential dead cells with a high % of mitochondria. So as we can see here, before any batch correction we do, there could be some heterogeneity or differences inside these samples or these batches already, like the quality control matrix, they are quite different among those samples. So before we go in and observe the differences, here we just want to briefly introduce some of the key metrics that can be used to measure the batch effects. So the popular ones are the KBAT, LISI, ASW, and PCR, the principal component regression for the PCR. So KBAT is one of the metrics that was designed or specifically for the single cell experiments. And it reports the rejection rates from cell subsets with the K nearest neighbors with local K cells versus the global, the whole data set of cells. It calculates the Pearson related chi-square statistics. And then the higher rejection rate, close to 1, will indicate more bias from batch effects. So we try to use KBAT for this experiment, but because these samples, it integrates the results from like 5 data sets. So here it didn't run successfully for us because the batch effects was quite strong, and the dropout rate for a lot of samples, they have a lot of zeros inside. So it is we didn't use this KBAT matrix at the end, and then the matrix itself may be problematic with very different cell types. On the other hand, there are also 2 popular metrics that people will use to access to check the batch effects. LISI, this indicates the effective number of labels present in the N nearest cells, which also was used a lot of times to calculate the diversity of the cell of the cells in the samples. So the ISI, it was calculated based on the summation of probability of a batch observed in the nearest cells, and the inversion of it tells us the effective number of the potential number of, for example, batches present in these cells. So the local here means it fits a Gaussian kernel to calculate the final number of LISI matrix. It does have some difficulties when the batches are very different sizes. So here it may return some of the biases if the PB is very different for different batches or samples. And for the ASW, the average silhouette width of cell group. So this is also, it was one of the original metrics that was used to calculate the cohesiveness of the clusters. So it measures how cohesive a sample to its own cluster compared to other clusters. And it indicates here average distances between the within group distances and the nearest neighbor distances. So represented in this formula here. The larger ASW, when it is close to 1, it indicates more bias from batch effects. So the next one is the principal component regression. So it is a PCA based, and it will tell the global effects. It calculates the PCA correlation with the batch variable with the principal component weighted by its variance attributes. So later, when we actually calculate the principal component regression, we break it down to the T or the major principal components and calculates the R square correlations that are basically the proportion of variance contributed to the labels from the input matrix. So the larger PCR indicates more bias from batch effects. And of course, there are other metrics, for example, the adjusted random index or the normalized mutual information. This can also be used when we have two sets of labels. For example, one set can be the batch, and a second set can be the clusters we calculated. So from here, you can calculate like how much overlap between the clusters you obtained compared to the batches, or the clusters you obtained compared to the cell types. So from there, it gives you some indication of how whether my clusters are driven by the cell types or they are driven by the batches. So among all these metrics, the KBAT, LISI, they are KNN based. They are faster. The ASW and the PCR, these two methods, they are PCA based, so they are slower to run. When using this these two metrics, it is possible, for example, to subsample your cells and to have an indication of whether any of the batch effect is very obvious or serious in your data set. So, and finally, regardless of the quantitative matrix, because they are basically numbers, and then they were these, okay, we can use them for measuring the batch effects, but then the number itself is not 100% indicative of the final quality of the batch corrections. One of the important perspective for checking the corrected data is still to look at the UMAP itself, and from the clusters or differentially expressed gene results to check, for example, how correct the batch effect returns and whether you are overcorrecting some of the counts. So here, before we do any batch correction, this is the batch effect inspections from the raw counts. As here, we show, for example, the first thing to take note of is like we firstly look at the UMAP here on the right side. So here shows the original UMAP that was calculated from a top 2,000 highly variable genes. And it was colored by, on the right side, it was colored by the cell type, and on the left side here, this figure, it was colored by the batches. So like the original uncorrected data, it shows actually quite some batch effects as the purple colored cells, as well, like they are the CD40 monocytes, and the yellow colored cells, they are CD60 monocytes. They are at very different locations, and like we can imagine later when we do a clustering, these two group of cells, they will be put into two different clusters, and then when we do the differential expressions, we will be actually doing the differential expressions between different batches instead of between different cell types. And here on the left side, like it also shows the batches are actually occupying locations in the UMAP. And one more coloring I think is also relevant here is it is colored by the sample metadata here. So the orange color here are all of the normal cells. So they can be normal PBMCs or normal bone marrows. And then the blue or purple colored samples, they are the stimulated or the control samples from a diseased patient. So as regardless of the batches, if we go and come in and look at the metadata, we can see the batches are splitting these two groups of meta samples with different metadata. Later, one of the objectives to check whether the batch correction algorithm is overcorrecting is still go back to the UMAP, for example, to check whether the diseased sample, there is any diseased sample that is left. And then the cells, they are grouped slightly differently with the normal samples because we may be expecting some of the biological or treatment differences within the sample metadata on top of the sample level batch effects. So we initially looking at this UMAP itself, we observe quite serious batch effects. And then regardless of saying, okay, we look at the marginal plot of the PCA, the different batches, they obviously locate at the different places of the PC1 as well as PC2. So this, at least these two PCs, they are heavily driven by the batches. And when we look at the PC regression, so the R square association between the PCs and is different phenotypes, the colors. So here, these two rows shows the proportion of variation that is explained by the cell types. So okay, we can see the proportion of the variance that is explained by the cell types from the original data for the first PC is not low. It is still okay. It's about like a 0.6, 0.6. But when we look at the percent of batches that is explained by the first PC as well as second PC, we can see that the percent of batches that is explained by the first PC and the second PC, they are as high as, for example, 0.4, as well as 0.8, where for the second PC, the cell type can only explain to about 0.1 and 0.02. So combining these, again, it is consistent with our observations. There could be some batch effects in this data set. And lastly, we look at the gene, the per gene level. So each of the red bar here is a gene. The gene level, per gene level correlation of the variance explained by the batches. So at the gene level, the variance explained by the batches, they are actually, for many of the genes, is very high, like 0.8 or above. So it means for this data set, we cannot reduce the batch effects by just removing the genes. We need to do a proper batch correction. So the next part, there are several options for batch corrections. So first one is like the normalization, log normalization, or the ST transform from CRET. They conduct a different normalization by summing up all of the counts of the same gene rates into the same amount for each of the cells, or regress out some of the variance, like the percent of mitochondria or the library size. And also there are other methods like regress out specifically and check residues with technical covariance, or remove those genes that correlated with batches. So in some of the many of the review papers, it mentioned that by normalization itself, or the regress out itself, and from some of our previous observations when analyzing the data, when the batch effects are very heavy, these methods are not enough to do a proper batch correction to return us the proper groups or subgroups of cells. So we still, most of the case when the batch effect is very heavy, we still rely on the proper batch correction tools to conduct the batch corrections. So for those batch correction tools, the typical method is like to select anchor or variable genes, or some tools will select anchor cells. and then conduct the normalization of those highly variable genes, and then conduct the PCA first, or conduct the batch correction first, and then it will return to the clusters. So let's go to the next slide. There are several, if we break them down, there are several different categories of tools that can be used for batch correction. The one of the earliest tools use the linear or the regress, more like the regress out methods, like for example, the combat, it used the it has the empirical Bayesian statistics behind, and then when using this tool, it explicitly modeled the predefined group of cells. So when this happen, it may miss the biology behind, like for example, in the previous UMAP, when we observed the disease samples versus normal samples, even for the same treatment types, they may exhibit some differences. So it is possible, like sometimes the combat may overcorrect with regard to this kind of situations. So in this example, we didn't test the combat, and also it does not work when there are many zeros in different batches. So the other three groups of batch correction tools are like more recent development, are anchor or distance-based tools, like the, for example, the MNN use the mutual nearest neighbors to do the batch correction, the CRET. On top of MNN, it adds in the CCA to calculate the reduce the dimensions from these MNN distances, and do a weighted nearest network calculations to map, to project the new data set to the reference data set. And then the Scarno-Rama used the SVD for the batch correction. Yeah, so for the anchor or distance-based method, it assumes similar cells exist in multiple batches, or it assumes there is a big training data set. It can be relatively slow, and then it is sensitive to the order of cells. Another group of batch correction method is the cluster-based method. So like both Harmony and Liger, they belong to the cluster-based method, where the clusters can be created and corrected using iterations of calculations. And the more recent ones are the deep learning methods, made use of a lot of these tools made use of autoencoders to try to infer the correct counts from the input samples. So there are at least a few sample tools. For example, map batch made use of the ensembled autoencoder, trying to infer the relationships between different cells after the autoencoder calculations. And so the deep learning method, it can represent the complicated nonlinear relationship behind the data. And then, like for example, the map batch, it projects to remove the batch effect, a batch specific effect, and then only the diagonal signals are kept, so that only the biology within the sample is kept, and then it avoids the overcorrection of the data, and it has enabled the discoveries of rare cells in the data set. And this is especially very useful when we have, for example, the treatment versus control samples. And then let's observe so the different tools, we here tested 7 different tools. And for each of the tools, most of them actually comes, made use of the highly variable genes, and then followed by the embedding, and the clusters can be obtained from the calculated embeddings from the tools. For the cluster-based tools, because their focus is on the cells themselves, so they don't return like the integrated assays, for example. So it returns more of the clusters from the tools. And for the deep learning methods, for example, it has the capability of returning both of the corrected comes from each of the genes, and it reports the embeddings, which will help to derive the proper clusters from the experiments. So let's observe the results we obtained from each of the tools. So the first tool we tested is the MNN. So it is a KNN-based tool, and then it tries to find the similar cells represented as mutual neighbors between different batches. So different neighbors here as connected by the dotted lines, they are assumed to belong to the same cell types. And then iterated steps of batch correction is done to make this correction. And then by projecting or finding these correction vectors, the direction of a test data set to a reference data set can be identified. And then when the projection happens, because of the directions from these groups of cells, multiple groups of cells, it helps to project not only the matched cells, but also the unmatched cells. And then this can be done iteratively when the new batch comes in. So the fast MNN was the MNN algorithm itself was implemented by in the CRET version 3 as well as monocle tree. So in the trajectory analysis, some of the trajectory analysis, as Adam introduced last time, the MNN was actually also used at the back end. So here we actually tested the fast MNN. It used the reduced PCA dimensions instead of the expression matrix, which improves the speed a lot. Okay, so now on the right side, it shows the result of the batch correction. As we observe, compared to the raw UMAP we had seen in the as previously, like the purple colored cell types, they were located at different locations from the raw counts, the raw uncorrected counts. Here, the cell types are grouped using a fast MNN. The cell types are grouped more close to each other by the same cell types, but not by the samples. So here, actually, the MNN is actually doing a good job by correcting the batches by overlaying the cells more closely to each other from the cell types compared to the batches, and here by overlaying the it with the disease versus the normal samples, we can see it still keep some of the disease samples that is located slightly different with the normal samples. So it correct, but in a relatively conservative way, but it doesn't overcorrect in this experiment. And when we observe the marginal PCA, of course, the marginal PCA looks good. The batches are well mixed together, and then the PCR, the PC regression here also shows like okay, now after the correction, the highest variance, they are now explained by the cell types instead of the batches. The next tool we tested is the CRET MNN and the CCA. So it identifies the cross dataset pairs of cell anchors iteratively, and when doing these corrections, identifying when it identifies the anchors, it also filter out the low score correspondence to avoid the errors. So this method actually both did the identification of the anchor cells, and it also removes the tries to avoid as much errors as possible, and by filtering this low score correspondence. And it is on top of the MNN, it adds in one more step of CCA here, trying to multiple steps of CCA, trying to identify the closely correlated cells. And then later these cells are merged by the the mutual nearest neighbor algorithms. So yeah, so here, as we observe from the right side, the Seurat MNN also did a very good job by grouping all of the same cell types together, and then the group of the same cell types, they are actually quite cohesive here, as they do form a very big clusters for the same cell types. And then those cell types, they are, when they are different cell types, they are very different from each other. And it keeps the relative locations, for example, of the near, the closer cell types like the CD40 monocytes, CD60 monocytes, and the dendritic cells. They are all grouped nearer to each other compared to, like, for example, the plasma cells and the B cells. They are grouped together. And then when we come in to see the batches overlay of the UMAP, we can see the batch corrections are actually quite successful here as well, because all of the batches, they are well mingled, mixed. And then, yes, we can also see this from the marginal PCA, like these different components of PCs, they are still splitted. There could be a small batch here represented by the orange color that are slightly, they may have a unique group of cells here. But here, when we look at the colors by the disease versus normal cells, we can see that Seurat may be overcorrecting a little bit here by like including the diseased as well as the normal cells all at these one cloudy locations. So later we will look at the clustering result to see whether we will be able to find any disease-specific clusters here from the Seurat. So, but from the UMAP itself, we begin to have some doubt here. Is it maybe overcorrecting a little bit here, because the disease-related cells, they are hidden underlying with the normal cells. But of course, the correction itself is successful. The cell type still is the dominant factor for the first few PCs, and then the batches, they are become less obvious for the first few PCs. Yeah, the next part, we also tried the scarnorama, and then it this is the method that make use of like a more calculated the nearest neighbor of shared cell types of paired data sets here. It try to find the nearest neighbors and then try to connect them and then link them together by using the randomized SVD using the hyperplane locality sensitivity hashing and random projection trees. So by using these, it also speeds up the calculation as well. And then so after identifying the mutually linked cells from the corrected data, it kind of stitch all of the nearest cells together and create unique distinct groups of cells that is represented in the clusters. So by using this way, the scarnorama itself didn't overcorrect. So it actually tries to remove the batch effects while remain the biology behind it as much as possible here by identifying these mutually linked cells using the SVD or hyperplane locality sensitive hashing. So this can be observed from the right side when we get the results from batch corrected UMAP, like the different CD40 monocytes, they are still located at different locations in this UMAP. And then when we look at the colors, we can see, okay, the diseased sample cells, CD40 monocytes, they are still stands out as from a different location with the normal cells. So this can also be observed from this 3-color overlaid UMAP here. Nonetheless, the cell type is still the most driving part with regard to the PCA, and then the batch effects, the batch are corrected here compared to the original uncorrected data. So it is still better than the uncorrected data, but then it is a little bit conservative when correcting the batch effects. The next one is Liger. So now we begin to move into the method that use more of the clustering-based method. So it made use of the non-negative matrix factorization, and the benefit of this non-negative matrix factorization is like the reduce the dimension here, or the factors that is learned, or the embeddings that is learned from this method has more biology meanings. So by single out or taking the genes that is calculated from this NMF, it is possible to derive, to look at the gene list itself and then find out the biology meanings behind it. So this is one benefit of using the NMF compared to the normal PCA calculations. So the input of Liger is it takes multiple data sets which share the gene level features. This method itself can handle the multi-omics data. Yeah, and then these cells, they are numbered by the, they are broken down here as a diagram. By using the NMF, so the blue color and the red color, they represents two different batches, and each of the batch, they are decomposed and then fit by a shared matrix together, shared factor embeddings, as well as the data set specific embeddings. So these shared embeddings will be the same among all of the batches, and the data set specific embeddings, which represented by the V in different colors here, they will remain different in each of the data set themselves. So by having this, the algorithm tries to find the shared matrix and then remove the differences in the data specific embeddings. So there are several parameters that you can use to calculate the Liger corrected data. So like, for example, the one of the important parameter is the K. K will equals to the number of clusters, and then the lambda will assess the balance between the factors across or specific to these batches, because the weightage of these two types of matrix are actually kind of important for this algorithm. And the next part is like it will calculate a joint clustering using the shared factor neighborhood algorithm that is from the Levin clustering. And then the cluster of the cells in the Liger is not dependent on the cell itself, but it made use of the neighbors, the clustering neighbors of the other, of the neighbors of the cell to determine the clusters of the cell itself. And then it is believed from this algorithm that they are more robust. Yeah, so the result of the Liger also shows like the batch correction was successful with regard to the cell types, but it's similar as CRET, it may be overcorrecting a little bit. So the disease samples are like hidden from the normal samples to some extent. Yeah, but again, the embedding calculations shows that the correction works by having a higher cell type driven variance compared to the lower part of the batch driven variance. Okay, the next part is harmony. Harmony is also cluster based, and then it did the iterative adjustment from the embeddings by creating. So here, like the data set is represented by different colors, and the cell type is represented by different shapes. By iterative clustering, it brings in the cells from different samples, same cell types together by regressing out the batch or sample specific variance in each of the clusters. Yeah, and they are, because you from here you have the beta variables, so you from the coefficients here you will learn a direction of where you want to correct these two. So and then from here you can always regress out or control this type of batch specific variations within the calculated clusters, and then this step is repeated until the converge is reached. So harmony is always like one of the fastest tool that is reported in the literature, and then it does the simple batch correction very well. As we can see here, like the cell type, they are very well separated. The CD40 monocytes, CD60 monocytes, and dendritic cells, they are very well located together, and the T cells and NK cells, they are the locations in the cell type UMAP, they make a lot of sense. And then the plasma cells, they are grouped together with the B cells. So so when we look at the batches, there could be slightly overcorrecting for the harmony itself. So, but still we can see like the green color disease samples, they may be dominant in some part of the clusters, but still it is possible to observe like a small group of cells that may only belongs to the normal cells. So yeah, we will confirm with the cluster result in the next few slides. The SCVI is, again, I think one of the more statistically sound method that tries to do the batch correction. It uses a combination of variational autoencoder together with the generative models, where it tries to impute the information from cell-specific library sizes, as well as the it tries to calculate the out of the other variants here by represented by the Z matrix. So in addition to the batch correction, it has additional capabilities of impute the differential expressions from the data. And when we look at the batch correction results, it seems the SCVI in this data set is a little bit conservative. And then similarly as one of the earlier tools, so the cells from different samples, they are still more or less away from each other using SCVI. So the next part is the map batch that we have seen. So map batch is developed by Dr. Chen Han and one of the close collaborator with Singapore. So it made use of the autoencoder trained samples. So here is a schema explanation of how the method works. Basically, in each of the input samples, all of the these two samples, for example, they are projected into a biology space S, and all of the orthogonal signals here are removed due to when calculating the autoencoders, and then we'll fit the data from this one sample from these samples. A clean embedding space is returned, and then it represents the cell type specific information within the sample itself. So no batch effects or no technology variation is learned from these autoencoders. And then the next step is to concatenate the learnings from like a small number of training samples and concatenate them together so that later when the test data comes in, they will always be projected to the, like for example, the 2 or 3 training samples, and then the corrected counts will be obtained. So the downstream differential expressions can be calculated from there. So this is a case study from Dr. Jung, where by using different other methods, like this is a simulation. So this is before correction data for the figure D, and then for the map batch, the result is in figure E. So a group of blue colored cohesive cluster can be observed after applying map batch. But all of these clusters or small variation of clusters, they were lost when using all of the other tools. And actually in our little small benchmark study, we observe exactly the same thing. So the map batch, it takes in the normalized rate counts, and then it calculates the top highly variable gene from each samples. It will output the ensemble embedding space. So it will tell us both the ensembled output as well as PCA output, and then it calculates the number of clusters and curate these clusters with cell types, and then return the UMaps. So it's a comprehensive toolkit that we can use for the batch corrections. So this is the result of map batch, and here we can see that the map batch, it works very well with regard to the cell type. It actually groups, like for example, the CD40 monocytes, CD16 monocytes, and the dendritic cells, they are all grouped here, and then they form different locations in the UMap. And then when we look at the overlay with the batches, they also located at a different place. But what is the best here is like when we observe the UMaps, the disease specific cells, they are part of them, they are embedded in the normal cells, and still there are some part of them, especially those small rare cells, they still stand out. And later when we do a clustering, we may be able to capture it. So it makes map batch a really good tool when you want to capture the small rare cells. Specifically, you want to capture something that has, for example, the prognosis powers with regard to the diseases. So we will actually highly recommend this tool, and we will try to find such things in your studies. And part of the map batch standard output, it will show the overlay, for example, of the samples with regard to the clusters. So here on the right side, each of the columns here, they are representing each of the clusters, and then the colors here represents each of the cells. So actually, we can see the map batch result, it is highly consistent with the cell types as shown in this UMap, and then it returns you the deeply curated cell types in clusters, as see here, like each of the clusters, after running the map batch, and with regard of another cell type annotation toolkit here we use in Single-Round Singapore, we are able to find the curated cell types here, that is deeply curated for those clusters, and then it does not overcorrect by still showing you the disease-related cells. And actually most of the the clusters, they are not batch driven. So it actually tells you it does the correction, and it does the correction very well, but then it does not overcorrect. It still gives you biologically meaningful results. So yeah, so here as you see the batch, the clusters, they are like automatically annotated once this program runs through. And again, it returns you the PCR results shows the high variance is explained by the cell types, and then it is less explained by the different batches. So when we combine it, when we group and show all of the the results from the 7 batch correction tools together, we can observe that for this specific sample, for example, mini benchmark, for example, it is the cells, they are relatively simple. They are all like PBMCs or bone marrows. So there is not like very complicated differences in this sample. And then the only small difference that happens here may be the disease samples versus the normal samples, these two disease samples that are different. So when we look at the LISI as well as the ASW scores, most of the tools actually they are not bad. They do what they are supposed to do by bringing up the LISI for the batches, like for Liger, for MNN, for Siret, for map batch, and it bring yeah. So and it didn't change much for the cell type. So the cell type remains as as one part of the important variation source. And it increased the ASW on the other hand increase, where shows more influences by the cell types, for example, compared to the batches. And again, also here we show that, for example, the CIRAT, SCARNA, ROMA, map batch, and Harmony. So these 4 tools actually perform relatively well when we compare with regard to the PC regressions. So yeah, so this is the general results here. And then I think I may be running a little.
Sorry to interrupt. You only have 5 minutes left of the hour.
Okay, sorry. Yeah. Because this tool is so good, so I really want to introduce this tool. And then yeah, I will speed up. So I have to apologize. Yeah, we will have the Q&A after the talk. So, but let's just quickly summarize here. Like when we look at the clusters results, like the results, for example, here, the small rare cell clusters from these disease samples, it does stand out, and then it did return the clusters here. So I think actually the map batch is really good to return this result. But on the other hand, some of the other tools may not be, may still be good. Like for example, if the data set is simple and you do not care, you like the differences between the disease versus normal cells, for example, are not of a very big concern. You can still use the tools like Harmony or the CIRAT CCA. They still give you cohesive results. It's just the CIRAT CCA may run a much longer time compared to Harmony. So we still recommend using Harmony when your data set is big. So if you have a big data set, we still recommend Harmony over the CIRAT CCA. Right. So yeah, so it's just to summarize, like each of the tools have different pros and cons, and we will highly recommend map batch if you are trying to find those rare cells in between different conditions of the metadata of the samples. And there are, of course, different challenges of different batch correction tools, like how to capture the diversity, how to handle the multi-omics, how to source for the data, and then whether it gives you what about the computational resources. Yeah, I forgot to mention that actually the map batch itself, because it only needs to train a few training samples, so it will not occupy a lot of computational resources. And then like whenever you have a new sample, you can just project it on the existing training samples. You already have a very good result, and then it's actually quite computationally scalable. So I'll just go, I don't think I have the time to go through the use case. It's just to let we know that, like for example, this is a real study on the real patients using this map batch tool. And then it finds out that the cluster 26, it is more details can be found from the Dr. Yung's poster published earlier last year. It actually is able to find this cluster 26. It is able to give you prognosis with regard to the survivals. And then it is the only cluster that is correlates with the PFS. So you are really getting whatever you want with regard to the disease progression. And then these cluster, as seen, I think from the previous mini benchmark, give you the cluster that may be lost from the other tools. Yeah, and then furthermore, it is not only possible to, it's not only you can find it from your data set, you can also re-identify the clusters in the new data sets. So this method, when you have these clusters, it is also possible to extend to the new data set. You can find a similar group of cells using the same method. So it's extendable. Yeah. Okay. So if you are interested, we may have a second advanced seminars where we can invite Dr. Yung himself to talk a little bit more about the map batch and how to make a good use of it, and maybe more user cases from there. So I will skip this summary, but just the short summary. So again, the Harmony or CIRAT itself is a good tool, but if you want to use the speed up the process, the Harmony is still good for the fast integration. Map batch is a great tool. It's highly scalable, and then it helps you to find those rare cells, especially with regard to disease progression. And then that's the key takeaway. So thank you. Thanks. Special thanks for Dr. Jonathan and Dr. Cheng Han, who made this most of the materials of this presentation. And thanks for Singapore office for proofreading of these slides. Thank you.
Thank you so much.
Thank you, John.
I don't think we have any time for questions and answers, but I said in the chat, if you do have any questions, just write it in the chat and then we can email you the answer later. So I just want to thank Stacey again for the really fantastic talk, and thank you all for attending. So I'll leave it open for a little bit if you have any questions, but other than that, thank you.